Me too.
All right, good afternoon, guys. I trust everybody is doing well. Kindly indicate to me if I'm audible enough and you cannot hear me.
Please, we can hear you.
If you can hear, we can hear you.
We can hear you. You can hear you.
Hear you.
We can hear you.
Yeah, we can hear you.
Huh? Ada ya, sisi sama saudara.
All right, it's great you can hear me. So, for obvious reasons, I think let me retreat the ground rules again. All standard rules for class engagement apply.
Hey.
Guys, hold on. Yeah, I'm trying to share the screen, so give me a little bit of. I can hear you.
Please, I'm here with a friend. She's on no group.
Hold on, and let me. Yeah, I wanted you to make me a host, but it's okay now.
Just took opportunity, but there's conflict and there's good.
All right. Somebody was indicating that a friend was with her. Can you please come again?
Hello, sir.
Yeah.
Hello. I was saying that Okay.
Yeah, Rosina.
Rosina.
Hello.
Yeah, I can hear you. Go ahead and speak.
So I was saying I'm here with a friend. She's from Joe. And she's also
Wilhelmina
facing the
Ojo.
same problem. So like, yes, right now her connection is off. Like we are using
Hello,
each
sir.
other. So whoever holds connection is able to use that one. Like in the meantime, do you understand what I'm saying?
So Rosina and Wilhelmina Ojo. So I'll take
Yes,
notice of that. Yeah.
yes.
Hello,
And
sir.
then so I wanted to ask a question.
Go ahead.
Okay.
Hello.
One person at a time. So please wait for the Christabel. Please wait. Hold on. Yes, Rosina. Make it snappy.
Yeah, the degree of freedom. All I heard was n-1.
I'm saying that I
And
take
then
notice of
I
that.
kept
So
on hearing,
hold on. I'll get there again. Christabel.
oh, okay.
Hello, sir.
Yes.
Sir, please. I'm also here with a friend. She's got down sir.
Turn on your video and let me see her. Christabel,
Please,
quickly
yeah,
now.
yeah. Can you see us? I don't even know. The video is more. The video
We can't see you.
Sir, please, I'm here, but I don't know why the video is more.
So when I said turn on the video, you said H.
But we are here.
If she's with you, find a way to get on your
Sir,
video.
no. We've seen the video on, but it's like.
Let me read to you. So if you want to prove that somebody is with you, you should be able to show that the person is with you. I can ask you to share a video. If you're unable to do that, I don't think there's anything else you can do about that again. Mami Ajoa.
Yes, please, sir. Please, I'm also here with a friend. I'll give her.
So where is
So
Ogiva?
please, we are turning on now. Yeah, she's here. I'm turning on the camera, please. So,
All right. So next time you guys should just put that in a text space. I'll observe that later, and if I want a proof, I would ask for it. So let's move on with the class. All right, so this is a question you are supposed to reflect on. But earlier on, I told you that three things will be important. First, you should be able to calculate the chi-square value, which is pretty much easy to do. You need the observed values, you need the expected, and then you can be able to computate that arithmetically. Second is to know the degrees of freedom. And the degrees of freedom is the number of categories you have minus 1, which is to say n minus 1. So if you are dealing with a situation where you have two different categories possible, you are dealing with, say, a character height, where there are two different options. That is to say, typical monohybrid cross, where two options of phenotypes are possible. Then your degrees of freedom are 2. Your number of categories are 2, and degrees of freedom is 2 minus 1. That gives you 1 degree of freedom. If you are dealing with a dihybrid cross, where you have 4 different phenotypes expected, then the degree of freedom is 4 minus 1, and that is 3 degrees of freedom. So however the number of categories, you subtract 1 from that, and that is your degree of freedom in that particular important. So we are supposed to consider a case study involving Drosophila melanogaster, and Drosophila has been studied extensively for genetic works for a number of reasons, including the fact that its genome is quite simple. It's very easy to cultivate them, to breed them. They are they have a very short life cycle and generally very advantageous in using them for genetic experiments. So you keep on seeing them again, over and over again. And notice that Drosophila melanogaster is also known as the fruit fly. So we are considering 4 different things. In Drosophila, the gene that affects the shape of the wing can be one of 2 types. If it's a C plus gene, it will produce a normal wing. If it is a C gene, it will produce a curved wing. Then the genes that affect the body color. If it's an E plus gene, it produces a gray color, and if it's a E gene, it produces an ebony. So notice that here we are having a typical dihybrid situation where we are considering 2 traits at 2 different characters at a time. We are considering wing shape and then body color. And for wing shape, you have 2 types of genes. C plus gene will be responsible for producing normal wing. C gene will produce curved wings.
Please say what's ebony?
With respect to body color, the gene for body color, you have E plus producing a normal color, and then E producing an ebony in that particular regard. Now, you're supposed to note that the wild type allele is the one that has the designation of the plus. So the C plus gene and the E plus gene are the wild type. And the recessive mutants are the alleles that are designated with the lowercase letters. So that information is particularly important. When you say the mutant gene, the mutant allele is the one that a mutation has happened in and is deviating from the normal, and it's actually a recessive gene, as opposed to the wild type allele, which has a plus, and that is dominant in that particular regard. So this is the information we have given this preamble. A cross is made between 2 breeding flies with the genotypes C plus, C plus, E plus, E plus. That is to say that this individual with the genotype C plus, C plus, E plus, E plus has normal wings and normal color, which is gray. So it has a gray body color with normal wings. And then an individual that is CCEE. So that individual has curved wings and ebony color. Now, the flies of the F1 generation were allowed to mate with each other and produce an F2 generation. And whenever a situation happens like this, realize that if it were to be a monohybrid cross, we'll be expecting a phenotypic ratio of 3:1 in the F2. If it's a dihybrid cross, what's the phenotypic ratio in the F2 that you should be expecting? You should be expecting 9:3:3:1 as a phenotypic ratio hypothetically. So that is the indication that should come from your head in theory. If it's a monohybrid cross, we are talking about F2 producing different variations. In the F2, phenotypic ratio is 3:1 in monohybrid cross. But with this being a dihybrid cross where we are taking 2 traits at a time, phenotypic ratio for F2 is 9:3:3:1 on the F2 ratio. All right. So this is the information they've given, that the F2 were allowed to mate with each other to produce the F1 were allowed to mate with each other to produce an F2 generation. And then this is the outcome of the mating. in the F1, all of the offspring had straight wings and gray bodies. And it's importantly easy to understand why that is the case. That in the F1, all of them had straight wings and gray bodies. Because the gene C plus that is responsible for the normal wing, which is straight wings, is actually the dominant gene and is a normal gene. And then the recessive mutant is a C. So if that C plus is dominant, then all of them indeed are going to show normal wing in F1 because dominant will triumph over the recessive gene. And then the E plus, which was responsible for gray color, is also dominant. So if a C plus, E plus, E plus mates with a C, C, E, E, then obviously all the offsprings are going to show the dominant traits. That's why in the F1, all of the offspring has straight wings, which is controlled by C plus gene, and gray bodies, which is controlled by the E plus gene. Now F2, this is what we have. After all the offsprings in the F1 are allowed to mate with themselves and produce in the F2. In the F2, with a total of 352 flies, we have 193 straight wings gray bodies, 69 straight wings ebony bodies, 64 curved wings gray bodies, 26 curved wings ebony bodies. Now you can do a little bit computation. Now 193 is to 69 is to 64 is to 26, and divide everything by the smallest number or a common factor and see if you can get the ratio of 9:3:3:1. If you do, you can get something close to that. But you can realize that there are deviations. For example, you have 69 here, you have 64 here. Meanwhile, they are all supposed to be 3:3. There's a difference of 4, a difference of 5 here. What we are going to do with the chi-square is to prove whether these kind of differences we are observing in themselves are very significant differences to form a grounds to reject the result from the experiment, or we can accept these differences or deviations within the range of experimental errors. So in applying the chi-square test to this, first thing is to propose a hypothesis that allows us to calculate the expected values based on Mendel's law. And if we take the assumption that the two traits are independently assorted, then using the Mendelian ratio of 9:3:3:1 on F2 generation, which is a typical Mendelian phenotypic ratio for F2, 9:3:3:1 for a dihybrid cross, the total ratio will be 16. That's 9 plus 3 plus 3 plus 1, which is 16. So we will take each of the options 9/16 times the total number of flies that we have in the F2, which is 352. The total is 352. So 9/16 times 352. The expected number of flies you're supposed to get based on Mendel's ratio is 198. So we are supposed to have 198 straight wings and gray bodies. So this is the expected number of flies with straight wings and gray bodies. However, let's look at what we got in the actual experiment. In the actual experiment, we got 193 straight wings and gray bodies. What's the difference? A difference of 5. Chi-square test will be determining whether that difference of 5 is statistically significant or not, and whether that forms a grounds to accept or reject the results from the experiment. If you do the next one, 3/16 times 352, that one you get 66 expectation of straight wings and ebony bodies. 3/16 the same 66. But if you look at the data that we have from the actual experiment, we don't get 66. Instead, we get 69 and a 64. And then the last one, 1/16 times 352, we get 22. Expected value of 22, but the observed value from the experiment in itself was 26 instead of the expected value of 22. So the first thing is to use the ratio, which is the theory, calculate from the total and get the actual expected numbers. So you know that the ratio is 9:3:3:1. So first one, 9/16 times the total, you get 3/16 times the total, you get, and all that. And then you get the expected numbers. Now, the observed numbers have already been given to you in the question. You use the ratio to obtain the expected numbers. So now you have your observed values, you have your expected values. You're on a good step to calculate your chi-square. So this is what you do. From the formula of the chi-square, this formula, S squared equals sigma observed minus expected all squared. You take each observed value minus the expected, you square it and divide by the observed. And because this summation, you add for all the categories. It's a summation of all the categories. So you do for straight wings, gray bodies, plus that of straight wings, ebony bodies, plus that of curved wings, gray bodies, plus that of curved wings, ebony bodies. You do for all of them. So let's see what we have. In the first instance, you have 193 minus the observed was 193, but the expected value was 198. You square everything over the expected value of 198. You do that for all of them. So, for example, those with straight wings and ebony bodies, expected value is 66, but the observed value is 64. No, 64. Straight wings, ebony, the observed value is 69. Straight wings, ebony bodies, observed value is 69, but expected value is 66. So let's see how that computation went. 69 minus 66 all squared all over 66 in that way. So you add everything, you add all the answers, and the chi-square value that we get here is 1.06. So the chi-square for the data that we have, given the observed and expected, is 1.06. Is there any question at this point before I continue? Is there any question, guys? Dr. Chelsea.
Yes, sir. So please, what is the interpretation of the chi-square?
We are yet to get there. We've gotten to a point where we have the chi-square value. We'll get to a point where we get to tell what it means and what we should do. So I'm asking that if anybody has a question up to where I've explained to. Yeah. Richard Owusu Boateng, doctor.
So my question is, how many decimal places do we use?
From your elective mathematics, how many standard decimal places do you have?
3.
4. It's 4.
Say 4.
So for me, my maths teacher has always been insistent on the fact that you leave your answers in 2 decimal places or 4 decimal places. I hope that's fine.
Yeah, right.
2 decimal places or 4 decimal places.
Yes, sir.
3 decimal places is not standard.
Yes.
All right. In the absence of any question, I'll move on from where we have our chi-square value and actually coming to tell what that means. Rosina.
Yes. I wanted to ask that instead of going through this long distance, why not draw a table system?
Yeah, you can actually use a table as well. But at the moment, I can present, this is what I have in the slides to present. But it's also been noted that using a table is also possible and quite simpler. But there is no means for me actually to draw the table right now. But if you've already seen how to do it on the table, that's still fine. but you should be rest assured that any of the methods that you should give you the right answer.
Thank you.
Okay, so I guess that's all with the concerns you have up to now. So I'll continue from here. Now, this chi-square value has a story to tell. And the story that is carried by this chi-square value of 1.06 will be best told by going to know what it actually represents by making reference to a statistical table. It does not rest within anybody's assumption to interpret this unless we refer to a statistical table. And notice that if should you be given any question by anyone, you'll be given a statistical table, which is an exam, so you can make reference to. We'll print out that statistical table for you to use. So step 4 is to interpret the chi-square value. The calculated chi-square value can be used to obtain probabilities. That is to say, p-values from a chi-square table. And these probabilities would enable you to determine what the likelihood of observed deviations are with respect to the random chance. That's who is speaking? Please be reminded that you keep mute at all times unless you have a question to ask or you have a contribution. So what I'm saying is that chi-square value of 1.06 that we have can be used in obtaining p-values or values of probability from a chi-square table. And these values of probability would enable us to estimate the likelihood that the differences that we are having in the values occurred out of pure random chance, and that our results happen out of pure random chance, or they're actually within the range of acceptable experimental errors. In all of these considerations, recognize that a very low chi-square value would indicate a high probability that observed deviations could be due to random chances only. And a high chi-square value would indicate that there's a low probability that observed deviations were due to random chance alone. Normally, if the deviations are due to random chance or very minute sampling errors, we tend to probably be lenient or lenient about it. But if the deviations are not as a result of random chances alone, but by serious experimental errors, for example, errors with the setup of the experiment or the experimental design itself, then it becomes very difficult to accept such a result. And once again, be reminded that if the chi-square value is high, it indicates that there's very little probability that the deviations you're having is due to just experimental, is just due to pure random chance, but something else quite significant. And if the chi-square value is low, then there's pretty much high confidence that and high probability that the observed deviations are by just pure random chance only. And we cannot understand what it means to say high or low unless we have a reference point. And the reference value is what we are going to see on the chi-square table. So if our chi-square value, if the chi-square, now notice this point very well. If the chi-square value that we have calculated, in this case 1.06, tends to be higher than the value that we will read on the table for the given margin of error and degrees of freedom, if our chi-square value is higher than that value, then we reject the null hypothesis, we reject the results, and we will not accept the results. However, if the chi-square value that we have calculated is lower than the value you obtained from the table, then we accept the result, that the result is quite valid in that particular regard. So let's move to the table and see. Now, notice that Once again, 4. If a chi-square value results in a probability that's less than 0.05, that is less than 5%, the hypothesis is rejected. And the hypothesis here we are talking about is the null hypothesis. And notice that in research, we have two types of hypothesis: the null hypothesis and the alternative hypothesis. Null hypothesis, like in very convincing words, is to say it's a prejudice against the results that we have. And where are the grammarians? When we say there's a prejudice, what's the meaning of prejudice? Like, so we get to understand what exactly is going on very well. Prejudice, what's the meaning of that? Any grammarian to help us? If not, I'm going to mention names, and I'll mention somebody who attended UAS. Let me find out UAS boys. You can best assist with these things. Hey, it's very randomly. Let me just mention any guy. Dr. Osei Richard Bodi. Prejudice. Okay. Okay, Evie is not getting on. Abdul Wahab, tell your roommates to turn off the music.
Okay.
And if you are the one playing the music, to turn it off. You are in class, for crying out loud.
Oh, please, I'm not the one. The music is near me. I'm not the one.
So are you in
please,
your room?
it means, yes, please.
And can you plead for your roommate to be civil a little? You are in class.
Okay, sir. And like, it's an opinion that's made without any reason or like without any experience.
It's an opinion that's made without reason or experience.
Like not based on any reason.
All right, really a dictionary consultancy.
Assumption without
that's great.
Without
That's
reason.
great. Thank you very much, Dr. Richard. Dr. Robinson Amwafo, are you an OWAS boy? Dr. Robinson, you can go ahead. You're an OWAS boy?
Yeah.
Oh, that's great.
Yes, sir.
So let's
so prejudice is something, so it is not based on natural experience. It's like something that you've seen, not actually experienced.
All right. And thank you very much. And it's important to note that normally things that are prejudicial are things that are unfair to the person that you're having a prejudice about. So, for example, the prejudice that all Kumasi people have a problem with their R and the L. You can have somebody from Kumasi who is pretty much okay with pronouncing those words. Or the prejudice that all black people are violent. Or the prejudice that all white people are not strong. So those things like that, they are not based on any concrete evidence about the individual. It's just a pure random assumption, which in case can be very unfair to that person. So when we say a null hypothesis is a prejudice to the results, what we mean that the null hypothesis is making an unguided assumption against the results by default, that the result is not accurate. That's what basically the null hypothesis is going to be. The alternative hypothesis would be the opposite of the null, and that will be that the differences that are occurring are not significant and that the result is okay. Now, if we reject the null hypothesis, we accept the alternative hypothesis. And if we accept the alternative hypothesis, we reject the null. So in that way, if the null hypothesis, which is a prejudice against the result, is maintained, then the alternative hypothesis would be rejected. If the null hypothesis is not rejected, then we would do the opposite to the alternative hypothesis. I still have some hands up. Joseph Papu. Dr. Joseph.
Say.
Yeah.
Say. Can you hear me?
Yeah, I can hear you.
I didn't hear the null hypothesis, the explanation. Please come again.
Okay. That's fine. Someone else had their hands up. Okay, in absence of any other question, let me clarify things again. I think Dr. Apafu had raised indication of that. What I'm saying is that the null hypothesis is a prejudice against the results. That makes an assumption that the differences or deviations that are occurring, differences from the result and expected values, is significant enough to make the results not valid. The alternative hypothesis is opposite of the null, which says that the differences that are occurring are not significant. Now, at the end of the day, from a chi-square test, we have to make a judgment or a decision, with which decision will be either to accept the null hypothesis and reject the alternative hypothesis, or we would accept, we will reject the null and accept the alternative hypothesis. And anytime we reject the null hypothesis, it means that the result is quite valid. And we reject the null hypothesis if the chi-square value that we have calculated is lower than the value that we read from the chi-square table using the degree of freedom and the margin of error. We reject the null hypothesis and accept the alternative hypothesis in that case where the value we obtain for the chi-square is lower than the one we obtain from the table. Another time, we accept the null hypothesis and reject the alternative if the chi-square value we calculated proves to be higher than the value that we read from the table. Then we would accept the null and reject the alternative. And notice that whenever we accept the null hypothesis, we are ultimately not accepting the results to be valid. And that accepting the null hypothesis is to agree that there is a significant difference between the observed value and the expected values. And so the result can't be accurate. Are you okay now? Joseph?
Yes, please. Yes,
is there
please.
anybody with a similar
Thank
concern? Dr. Nanajua. Those of you playing music in your room. Abdul Wahab.
Abdul Wahab, you have to mute your microphone.
So Abdul Wahab, how can you be playing this music in your room and still be listening to a class at the same time?
I'm sorry, sir. Sorry.
No, like I'm being curious about how you do it. How do you do it?
50 billion mark.
Well, that's a 10 100 billion.
100 billion.
Yeah, when they play tracks for you, we said, but you say how the person they play tracks are you so.
Dr.
Yes, hello, I have a question.
Okay.
My question is with regards to the very first lecture, you said that we do not accept the null hypotheses, but we fail to reject it. But then you just said that we can accept it. So is there, does it matter if we state that we fail to reject it or we accept it, or they're the same?
Nanajua, this is the issue. Inappropriate parlance. Hey, who is that? Elvis. The 2,000 marks. Now listen. If anybody unmutes themselves when they have no business contributing or asking a question, I'll just remove you. And notice that when I remove you, you can't join the class again until this maybe next week. All right. So Nanajua, inappropriate parlance, using the right expressions, We don't accept a hypothesis in statistics. We reject the hypothesis or we fail to reject. All right. I use the word we accept. So the last time I told you guys that I use the word we accept now to make it quite easier for people to appreciate that. And in any case, nobody would really concern themselves with the business of checking appropriate statistical diction in a genetics class. All that you will require is for you to understand what a chi-square value means and be able to make appropriate interpretation. Yeah, but also in your statistics class and all that, notice that it would not be right to say you accept the hypothesis. You instead say you fail to reject it. But conclusively, failing to reject and accepting are the same thing. But there's a reason why you don't say you accept it. You fail to reject or you reject it. Yeah. But in any case, failing to reject is implying that you accept it. Nanajo, are you okay?
Thank you.
Yeah,
Yes,
so let me
thank
use
you very
the
much.
definition. All right. Dr. Matthew Boateng.
Say I'm lost. Like, can you go over the whole thing? Like, I don't know where I am right now. Like the null hypothesis is that thing.
Doc, have you been connected throughout, or you got lost at the?
Yeah, I was listening to you. Degree of freedom, and everything. But like right now, what you just said.
What I just said in the last moment, that's what you don't get. If that's the case, I can just go over that one. That's not too much to ask for. But if your indication is about the whole show, then I'm afraid that could be too much to admit.
No, it's just what you said like in the past 5 minutes.
All right. Elvis, this is your last warning. Apart from the fact that you are distracting the class, the music you are playing is also not interesting.
It's not me. It's not me. I'm playing music.
Okay, let me get back to business. So guys, these things are not difficult, but they are quite technical. If you don't pay attention a little bit, by the time you are back, you are lost. So I'm at the point where you have to interpret your chi-square value. After you are done calculating the chi-square value, what next? In this case, our answer for the chi-square is 1.06. So we have to make an interpretation. What I've been saying is that the chi-square value we've calculated allows us to obtain values of probabilities or p-values from a chi-square table. If we get to the chi-square table, we need two things. We need the degrees of freedom, which I've said is the number of categories or options we have minus 1. So in a typical monohybrid course where you have two different phenotypes, your degrees of freedom is n minus 1, which is the two different phenotypes you have minus 1. And so your degree of freedom is 1. In a dihybrid course like this, where we have 4 different phenotypes, the degree of freedom is 4 minus 1, and so we have 3 degrees of freedom. You need that value for the degree of freedom in reading from the chi-square table. And then secondly, you need the margin of error or the confidence interval you are using. I told you that in most scientific works, if the question doesn't state, read it from 0.05. If the question otherwise states, then read from, if the question doesn't state the confidence interval or the margin of error, use 0.05. But if the question states otherwise, you have to respect the rubric of the question. For example, the question says, read from 80% confidence interval, that is to say 0.2 margin of error. That is what you're supposed to use. But this would ultimately become clear when we get to the table. So let me try and get a chi-square table. This is an example of a chi-square table. Guys, I hope you can all see.
Yes, please.
All right. Now, for this being a chi-square table, you can see on the top horizontal rows, and those who don't know the difference between rows and columns, row is horizontal, and column is vertical. So on the columns, you see the first column, you have 1, 2, 3 up to 30. Those are the degrees of freedoms. And then on the horizontal, first horizontal row, you have p values. So first you have p is equal to 0.99, 0.95, 0.80, 0.50, 0.20, 0.5, and 0.01. Who doesn't get this up to this point? Like just what I've said about this table, you don't get it. Let me go over again.
Me. Sir, me, I don't get it.
What don't you get? Is it I didn't see or?
I also don't get.
Sir, please go over.
Hey, Meshumo.
Sir, please go over. Why you?
I also
Sir,
didn't get
please
it. I
go
didn't get
over.
anything.
I didn't
Sir,
get
please
it.
go over.
Sir, please go over.
Please go over for
I
that.
beg.
It's okay. It's
Sir,
okay.
please go over.
Hey,
Sir, please go over. I beg.
HB1,
Sir,
not expire. What
sir,
do
please
you?
go over. You beg. Please go over.
So you guys should pay attention. I'll go over for the last time. So I've been wanting you to get two things from the table. Can you see the pointer?
Yes, sir.
So
Yes, sir.
the first
Yes, sir.
vertical column, the numbers there. You see 1, 2, 3 up to 30 on the first column. Those values are the degrees of freedom. So degrees of freedom 1 to 30 on the first column. Just in case you don't know what column is, a column is a vertical, and then rows are horizontal. Now let's come to the first row. The first row where you have the p values. You have p equals 0.99, 0.95, up to 0.01. So all that I've wanted you to pay attention to are the degrees of freedom and the p values. So there's absolutely nothing not to understand at all, unless of course you didn't see what I had wanted you to see. Now, since all of you can see this, notice that if I'm using a confidence interval of 95%, that is to say that I'm allowing a degree of freedom, or how do you call it? I'm allowing a margin of error of a margin of error of 5%. That 5% is 5 over 100, and that is 0.05. That will be this value here. So in that case, you should be paying attention to this p value of 0.05. That is if I'm using a margin of error of 5%. So that 5% margin of error is 5 over 100. That's 0.05. You should be paying attention to this margin of error. And then you should be looking at how many degrees of freedom. In the work that we just had, how many degrees of freedom do we have? We are having a typical Mendelian dihybrid cross with 4 different phenotypes in the F2 generation. That is to say that given 4 different categories, degree of freedom being the number of categories minus 1, that is 4 minus 1, our degrees of freedom are 3. So we have 3 degrees of freedom. So you trace from the number of degrees of freedom, that's 3 degrees of freedom, you trace up to, and for example, if we tell you to read at 0.05, you trace up to where you have the 0.05, and that value there is, for example, is 7.815. That is the value you should be paying attention to, and that would be the value that you'll be picking from the table. That is 3 degrees of freedom and a confidence interval of 95% or a margin of error of 0.05. This value of 7.815 is what you should be picking. However, if they tell you to read at, let's say, 0.8, they ask you to read at 0.8, then, for example, 3 degrees of freedom, that value here is 1.005. You'll be reading from here. If they tell you to read from 0.5, you'll be reading from here. So whatever degrees of freedom they ask you to read from, that is where you notice the degree of freedom, you trace up to the column that has the margin of error, and then you read the value there. It's as simple as that. I believe everybody should get this. If there are any reasons why you don't get this, including issues of color blindness and all of those things, kindly indicate to me. Dr. Dimi, you have an issue?
So please just to get this straight. So we are to accept the null hypothesis when the calculated chi-square value is less than the critical value at the particular probability, or we reject it when the calculated chi-square value is greater than the critical value.
So, Dr. Dimi, get this. You notice that the assumption that is made by the null hypothesis is not really good for our result. It's a prejudice against our result. So whenever we are rejecting the null hypothesis, it means the result is fine. If we accept or we fail to reject the null hypothesis, that's maintaining the null hypothesis, it means the result is not fine. You get it? So anytime the the null hypothesis is lower than the critical value, the chi-square is lower than the critical value, then we reject the null hypothesis. That means the result is fine. So if your chi-square is lower than the critical value, the result is fine, and you reject the null hypothesis.
Let me get this straight. So we reject the hypothesis when the chi-square value is lower than the critical value.
Yes. When the chi-square value is lower than the value on the table with the critical value.
Okay.
Are you okay now? Dr. Marina.
Yes.
I'll ask what Dr. Dimitri asked.
Okay, let's move on and see something. Now, in this particular work, they read from 0.80. And I've told you that the question doesn't tell you to read from anywhere. You read from 0.05. But here they read from 0.80. And they had a critical value of 1.005. 1.005. That's what they had from the table. And our chi-square value, just as a reminder, is 1.06. If you compare our chi-square value of 1.06 to this critical value of 1.005, that's reading from 0.8 with 3 degrees of freedom, then our chi-square value is a little bit higher than this critical value. That suggests to us that there is a problem with our results. If our chi-square value had been lower than this critical value of 1.005, then our results would be just fine. So let's see what conclusions we make, step 4 in interpreting the results. With a degree of freedom equals to 3 and a chi-score of 1.06, which is slightly greater than the critical value of 1.005, that is reading from p-value of 0.80. And note that a p-value of 0.080 means that values equal or greater than 1.005 are expected to occur 80% of the times based on random chances. Therefore, it is quite probable that the deviations between the observed and expected values in the experiment can be explained by random sampling error. So, in this case, there's admission that there is a problem with the results. Let me say, just maybe that problem with the results occurred by sampling error, how the researcher sampled the plants. There was an error in that, and so that is what is bringing the differences between the observed and expected. And that observed difference between the observed and expected value, according to the chi-square value and the critical value you obtain, is significant because our chi-square value is higher than the critical value. The critical value is 1.005, our chi-square value is 1.06, and so we, in this case, would fail to reject the null hypothesis. And by failing to reject the null hypothesis, we admit that the deviation is significant, and that this goes ahead to say that the deviations that are observed between the observed and expected values in the experiment can be explained probably by random sampling error. Yeah, but in any case, most of the times, your interpretation will just end up in you deciding whether the difference is significant or not, and that's whether the result is acceptable or not. So if your chi-square is lower than the critical value, then you reject the null hypothesis and that the result can be accepted. But if your chi-square value is higher than the critical value you read from the table, then you fail to reject the null hypothesis and the entire result is questionable, and so you cannot really accept it because the differences is quite significant. But you will not be obligated to explain why that differences is happening. This one just added that just maybe because of random sampling error that occurred. Any questions up till now?
Find the mean variance and standard deviation.
George Namiche.
Sir, with this question, why did they read from 0.80?
This question is an example you are being given in the notes, and I cannot be able to define why they decided, but it's theoretical. Do you get it? It's theoretical, so you could even say reading point 0.95. But I've told you that in any time you have a question, the question will tell you what margin of error to allow and where to read from, or if the question doesn't give you, use the standard one, which is 0.05. But I cannot divine why the question decided to say read from this. But it could just be an example. I could say, let's say if you are reading from 0. Let's say if we had read from let's say if we had read from 0.95, then the critical value would have been 0.352, and in that case too, I think that would have still been higher than our chi-square value in that particular regard. So you can read it from anywhere for teaching purposes, but in real life, standard scientific procedure for a lot of researchers, we read it at 0.05. But in any case, a question can demand you to read from anywhere, and you should pay attention to what the question wants you to do. Anything about chi-square analysis so far? Dr. Michael Kozu.
Sir. Sir, please. I actually learned a bit about the chi-square before this class on YouTube and on some websites. And what I learned over there was like, to accept the null hypothesis, the calculated chi-square value should rather be lower than the critical value.
To accept the result or to accept the null hypothesis? They are two different things.
So to accept the null hypothesis, I also came across that.
So to accept the null hypothesis, which is a prejudice against the result, the chi-square value should be lower.
Yes.
I think I have to further probably look at your source and all that, but a very credible source I also have is the indication that if the chi-square value is lower than the critical value that you have, then the result is acceptable, which in that case, the null hypothesis is being rejected. And notice that at all times, there's a difference between saying we accept the null hypothesis and then we reject we accept the null hypothesis and then we accept the results. They are opposites. If you accept the null hypothesis, which is a prejudice against the results, you can't be accepting that result. Because the null hypothesis assumes that the result is not, the difference that is happening is quite a significant difference, and so there's a problem with the results. So if you accept that, then there's a problem with the results and you cannot be accepting that result. So first, check if there is a conclusive mention of whether they were saying when the chi-square value is lower than the critical value, you accept the null hypothesis, or they were saying you accept the results. If they are saying you accept the results, I agree with them. They're saying they accept the null hypothesis, then I disagree. But because you are saying on YouTube, I'm quite unable to question that because it's not a very credible source you can reference. But is there anybody here who has seen that in a book?
Yes, sir.
Which book is that?
I use a Mary Jones.
Okay, so I think we would have to but apart from the same person, this is Dimitra, right?
Yes.
Apart from Dimitra, is there any other person who has seen a variable suggestion from a book? Not YouTube.
Yes, sir.
Who is that?
Richard.
Yeah, can you reference that for us?
From one of the slides I downloaded from one senior Google Drive, a senior course me. I read from it that the null hypothesis is the hypothesis that proposes that there is no difference between the expected observed.
Okay, do you have wait, let me get the slide on hypothesis testing. Is that in this same one or is another one? You guys have it, right?
It is an older one. Yeah.
Is in the other one, biometry. Let me get that quickly. Biometry.
Hello there.
Yeah.
Hello.
Yeah, who is talking?
Yes, sir, please. We also found that from Google that if your chi-square calculation is greater than the chi-square critical value, then you reject your null hypothesis for that.
If your what?
If your chi-square calculated value, that one is greater than the chi-square critical value, you reject your null hypothesis.
If it's
from
lower
what
than
you
the
were
critical
saying,
value, you accept.
it's less
All
than.
right.
Yes.
Okay, hold on a bit. Can you see the screen I'm sharing?
Yes.
Yes, please.
I'm looking at something here. Hypothesis testing. I think I'm not finding that, but one of you should quickly do this. Just search for null hypothesis and let me find something. If I get that, then I would consult Prof and then probably we make that amends. Can anybody do that quickly? Just search null hypothesis and then read what you have. Does anyone have it?
Yes, sir.
Please, one person at a time. Who is going? You just raise up your hand. I'm muting everybody, so you raise your hand and I call you. Dr. Steph. Steph, you should be on the floor. I want to clarify something. I think there's a little twist between the now and the alternative. If I get that, then you can quickly resolve it.
Okay, sir, I'm reading. Now, hypothesis is a type of hypothesis that is used in statistics that proposes that there is no difference between certain characteristics of a population or data generating process.
Okay.
For example,
that's what I wanted. So let's make that amend. So that clarifies it. So in this case, let's go with this. If the chi-square value is lower than the critical value you read, then you fail to reject the null hypothesis. And that to say that the result is fine. If the chi-square value you read is greater than the critical value, then you reject the null hypothesis and accept the alternative hypothesis. That indicates that there's a problem with the results. Is that fine with everyone? Dr. Dimi and Co.
Yes.
Yes, please.
Yes, sir.
I think the twist was between the now and the alternative. But so the explanation should be now inverted, that it's rather the alternative hypothesis that has the prejudice against the results, but then the null hypothesis makes a presumption that the result is fine and the difference between the observed values and expected values are not significant. So if that proposition by the null hypothesis that the differences are not significant, so if the differences are not statistically significant, the result is fine. And then the alternative hypothesis will be the opposite. They are always opposites. Then the alternative hypothesis will be that the differences is significant, and so there's a problem with the results. So once again, everybody is clear about how to calculate the chi-square value. When you get a chi-square value, now get your degrees of freedom. That is the number of categories minus 1. If you get your degrees of freedom, now get your p-value, where you should read from. If you are reading from 0.05, trace from 0.05 that column to the degrees of freedom you have and read that critical value from the table. Now compare the value you get from the table to your chi-square. If your chi-square value is lower than the value on the table, then you accept the null hypothesis, and then you accept that the result is correct. And notice that I use the word accept instead of fail to reject. Yeah. And then if you have an instance where your chi-square value is higher than the value you read from the table, then that ultimately suggests that you reject the null hypothesis and fail to reject the alternative hypothesis. That's accepting the alternative hypothesis, and there's a problem with the results. Is everybody okay now?
Yes.
All right, so Dr. Dimi and colleague, Thanks for that contribution. Should I dash 2 marks or 1 mark or 100 billion?
100 billion.
100 billion. Billion.
40 billion, 40
100 billion.
billion, 40 billion,
Trillion.
40
billion.
billion,
Um,
40 trillion.
doctors, when it's time for playing, I'll let you know. Let's continue. So your man with chance can clear. So we are done with chi-square. Let's quickly go to molecular genetics, the SC and Chase experiment, Avery, McLeod, McCarthy, and then we come back to pedigree analysis if we have time. So what do you guys think? You want pedigree first, and then the molecular genetics, or molecular genetics first, then pedigree.
Yes, sir.
So let's vote. As
I'll
many,
give it to
relax. As many people who want us to do the molecular genetics first, explain those experiments first. But I've got close indication that there are problems. Some people have problems there. If you want us to do that first, raise up your hand. Then we continue with pedigree. Raise up your hand. And so all those who want us to treat the molecular genetics, those experiments, raise up your hand. Quickly. I have 30 people now. What about overvoting? For this one, you can't do overvoting. Everybody can raise up their hands once. And some people want to vote twice. And those who are sitting with their friends using the same device, you can't vote. All right, I have 41 people. Hey, the number is still going up. The voting ends in 30 seconds. I have 50 people now. At this point, the number is still going up. We have 60 now. At this point, I think there's reasonable indication that I think you guys have reached the threshold. So let's do that first. But if I wait, a lot of people will still raise up their hands. So you should all lower your hands and then let's go to the next thing. So we'll do that. Let me get that slide. Are you guys all seeing the slide on molecular genetics, or you're seeing something different?
So we are seeing it.
I'm still seeing the error test.
I'm still seeing
You
the
are still
previous.
seeing the error
You're
because
seeing
you
molecular
have a
genetics.
latency in your internet connection. I think by now everybody should see the molecular genetics.
Yeah, it has come.
You can't see the, I don't know level of latency you have, whether you are still using espresso or something. All right. So Griffith experiment. I want two doctors to give us the bigger picture, broad overview of what a Griffith experiment was about. Just two people, like broad overview. 400 billion marks. Without which I'll mention names for 0 marks. Dr. Rosina.
Yes.
Yes, Ruru, go ahead.
Yes.
Mm-hmm.
Please,
Or you've not studied that one?
No, sir.
You've not studied that one?
But then I remember you have not studied it.
Hey, so what are you waiting for? You did that lecture on Wednesday, and today is Friday. Hmm?
Yes.
Yeah, so you tell us what you know.
When I studied, I remember Prof was saying it gives a medication that it will work on the person, but then it will work on another person, like something like that.
Hey,
That's what I remember.
Dr. Rosina.
Yes, yes, yes. Oh, but I
Okay,
studied it, okay.
you have not studied. It's obvious. Who can help? Dr. Dimi.
So please, the question again.
Is Renee in this class? Renee. Last week Renee was not here. Is he here? Is she here today?
I was there, sir, but I'm there, sir.
Are you here today, Renee?
I'm just having,
I think these days you've not
yes,
been
sir,
asked
I'm there. I'm just having disconnection every.
all the time. What network are you using? Use MTN.
Yes, sir.
I think these days you've not been answering questions, so it's become obvious you've not been around. Do you have a contribution?
I didn't get what the question, sir. Please, can you retake the
I'll just ask him for a
question?
brief overview of the Griffith experiment of transformation in bacteria.
Okay, so this experiment is simply about Griffith. He took he works with Streptococcus pneumoniae. So there were two strains of Streptococcus pneumoniae. The F strain, which is virulent, and the R strain, which is non-virulent. So he made experiments such that when he took that S.
Dr. Renee, which one do you say is which strain is virulent? Is it the S strain or the R strain? I think we lost Renee. Estelle.
Sir, is about the first experiment where we're using mice. So the type 3S is virulent bacteria and the type 2R is non-virulent bacteria. So when we inject the type 3S in the mouse will die because the bacteria is virulent. But when you inject the 2R, the mouse will not die because it is non-virulent. And now when you take the type 2R and heat it, and the type 3S and heat it and inject it inside the mouse, it will not die because it had been denatured. And when you mix both the type 2R and the 3S and heat it, some 3S will not be denatured, and this will lead to the death of the mouse.
Okay, thank you. Any other person? I'll try to get a video on YouTube. You can speak.
Sir, please, can you liken this whole process between the PRPC genes and the PRSC genes work for the prion disease?
I think that would be far-fetched.
Okay, sir.
Can you guys hear the sound? Guys, can you hear the sound? Can you hear the
No,
sound?
sir. No, we can't hear.
No, please.
It's okay. It's okay. It's
We
okay.
can't hear.
No, sir.
Frederick Griffiths made this experiment, and it's been called the Griffith's experiment of transformation in bacteria. And in this experiment, he took mice and injected them with strains of bacteria. And these bacteria were what we call Streptococcus pneumoniae. And these are the bacteria that are responsible for causing pneumonia in tetrapods in that particular regard. This bacteria had two strains, the smooth, which is the S strain, and then the R strain. And one of the strains was virulent. When you say it's virulent, it means that it's able to cause diseases. It's pathogenic. And that is mostly due to the fact that there is a presence of a capsule that is able to use to attach the host and all that. And that gives it that virulent ability. In this experiment, if you inject a mouse with the virulent strain it's expected that the mouse contracts pneumonia and dies out of pneumonia. If you inject the mouse with the avirulent strain, avirulent means the non-virulent strain of the bacteria, you're not expecting anything to happen in that particular regard. The broader concept of the idea of transformation as a principle in bacteria is that bacteria can be able to incorporate the DNA of some other bacteria within a colony, or even bacteria that are dead. They can pick up that genetic information, they can pick up their DNA and incorporate it into their own DNA and transform. And that's how come mutation in bacteria is at a very high rate and evolution in bacteria happens at a very prolific rate. Coupling the fact that they can incorporate extra DNA outside their cells into their own DNA and evolve, and also the fact that they can multiply quickly, bacteria can be able to evolve as fast as possible. So you have the type 3 S3 bacteria, which is a virulent bacteria, and then the R2, which is not virulent. So can you all see from the picture here, or you guys are still seeing from the video? Can you see from the slide, the picture?
No.
No.
We are still seeing the video.
All right, let me share the. Yeah, so which of the strains is pathogenic or virulent? Is it the S3 or the R2? The whole class.
S3.
S3.
All right, so in this experiment, the expected outcome is that if you inject a mouse with S3, they should die out of pneumonia. And that's a reasonable expectation. And if you inject with R2, they should survive. So this is what Grifith'S did. He injects a mouse with R2, which is the non-virulent strain, and the mouse survives. Then he injects a mouse with the S3. The mouse dies out of pneumonia because the S3 is virulent. Now, second, he wants to find out something. He injects the mouse with heat-killed S3, and the mouse does not die. The mouse survives. That is to say that by heating the S3 and it dies, it's not able to transfer or cause pneumonia, and then the mouse survives. So now we know that R2 does not cause any disease, and then S3 causes the disease. Who is that? We know that R2 is non-virulent, does not cause the disease. But we know that S3 causes the disease, is virulent. But we also know that if you heat kill the S3, it can't cause the disease, even though a living one would cause the disease. So now if we have a mixture of R2, which doesn't cause the disease, and then S3 that has been heat killed, which we don't expect to cause the disease, and we inject that mixture into a mouse, like under normal consideration, what should you expect? Because the R2 in itself doesn't kill. S3 that is heat killed doesn't kill. But if you add a mixture of them and inject in the mouse, are you expecting the mouse to die or to live? Under reasonable expectations.
To live.
Yeah, what do you think, guys?
To live.
To live.
Live.
All right, it's
Said,
okay.
I think the chance is
Now,
50-50.
anybody who believes under reasonable expectation that this mouse should die should raise up their hand. Is there anyone who believes that the mouse should die after being injected with heat-killed S3 and then R2? Samuela. Samuela, your hand is up. Marina.
Yes.
Marina. Yeah, one of you should speak. Why do you think so? Under normal consideration.
Circumstances. Because since it can play to the genetic material of the heat killed.
That's Dr. Marina, I think that's after you have this close idea of transformation in bacteria. But under normal consideration, a layman's point of view, if you inject with R2, the mouse won't die because it's avirulent. You inject with heat-killed S3, the mouse won't die. So how come when you mix the two, if you mix the two, everybody's expecting the mouse not to die. But mysteriously, the mouse dies after being injected with a mixture. Then the explanation is what Dr. Marina's tried to give. And the explanation is that the R2 bacteria, Streptococcus pneumonia, which is non-virulent, was able to pick up the genetic material or the DNA of the heat-killed S3. So even though the S3 was dead, its genetic material was still living. Its genetic material was still functional. So the R2 picked up the genetic information or the DNA of the heat-killed S3 and incorporated it into its own DNA, and that transformed the non-virulent R2 into a virulent S3. And so the R2 had evolved through that transformation by picking up the DNA of the S3. And at the end of this experiment, what really gave an indication to scientists was that the ability of bacteria to carry out certain functions is directed by something that can be transferable. So in this case, they realized that the R2 might have picked up something from the heat-killed S3 that has been able to change it to behave like the heat-killed S3. Now, the contemplation was, is that genetic information that is transferred from the S3 to the R2, was it DNA or it was protein? So now they are certain that something is responsible for that characteristic, that trait. And that something that is responsible was transferable from the S3 to the S2. Now the contention is that material, that genetic material, is it an RNA or it is protein or it is DNA? What further they did was, now in the mixture that they did, they added RNAs, and notice that RNAs is an enzyme that can be able to digest all RNA. So if you add RNAs to the mixture, you expect that it will digest all the RNA that is present in that mixture. And so even if an RNA was there, which potentially would have been transferred to the S2 and then given the virulent nature, since that RNA would have been destroyed by the enzyme RNAs, the transformation wouldn't happen. Then the second thing was also another mixture, they added protease. And these proteases are able to destroy proteins, digest proteins. So if you add that and that destroys all the proteins of the bacteria, then it is obvious that the heat-killed S3 cannot transfer its proteins to the living R2 such that it transforms. Then the next setup, they added DNase. DNase is an enzyme that digests or destroys DNA. So in that case, they've destroyed all the DNA. So if it's indeed DNA that was responsible for transferring the characteristics, then after destroying the DNA, no transformation should occur. But guess what? In the setup that they added the RNase enzyme that destroyed all the RNA, the transformation happened. That means RNA is not responsible for transferring that characteristic. Our RNA was not the ultimate genetic material, because even when they destroyed all the RNA, the transformation still happened. Then in the setup that they added protease, destroying all the protein, the transformation still happened. And that means that protein was not the true genetic material. But in the setup that they added DNase to destroy all the DNA in that sample, the transformation did not happen. That means that the DNA is the genetic material that was responsible for that transformation. And if you destroy that DNA, which is the genetic material, that transformation would not happen. And so that was what the Griffith's experiment was about. Now, after this, there was a reasonable indication that DNA is the genetic material. So the conclusion is this: because only the DNase destroyed the transforming substance, the transforming substance must be DNA. That is the conclusion that comes from Griffith's experiment. Now, scientists were still not really certain. Some people still maintain that proteins were the genetic material. Some people think RNA was genetic material. So there was a need for a conclusive experiment to end the discussion. And ladies and gentlemen, that introduces us to Hershey and Chase experiment in 1950. But before then, any questions with Griffith's experiment of transformation in Streptococcus pneumoniae? Any question with Griffith's experiment? So I'm going to ask a few questions. First question is, based on the results that they obtained, how were they able to show that DNA is the likeliest genetic material? And this question is going to be answered by Dr. Max Techi Isiama.
Okay, hello.
Yeah, Max.
Hello.
Max, go ahead. Everybody, the whole world is
Hello.
listening to you.
Hello, please, can you hear me?
The whole world is listening to you.
Okay. Well, in the experiment, okay, in the experiments, they added DNAs to the homogenized sample of the bacteria.
And
so
by inference,
they
homogenized sample means
injected it to the type 2R bacteria, and they realized that no transformation occurred inside it.
All right, thank you very much, Dr.
Hello,
Max. How they were able to conclusively determine that DNA was a genetic material is that when they added 3 enzymes to different samples, DNase, protease, and then RNase, it was only the one that contained the DNase that destroyed the DNA that did not show transformation. That's suggesting that that transforming substance might have been destroyed when the DNA was destroyed. And so DNA must be the genetic material. Next question. Why is it that the R2 is avirulent, not virulent? And this is going to be answered by Dr. Ama Sarisewa. Dr. Sewama, sorry.
Please, can you go on, please?
The question, Dr. Sewa, the question is, why is it that with the Streptococcus pneumonia bacteria, why is it that the L2 is not pathogenic, it's not virulent? The rough strain, why is it not pathogenic? And why is the S strain pathogenic or virulent?
Please, I think the R is the RNA and therefore could change the genetic sequence of the organism, whilst the S strain it contains DNA and
No,
could
no, 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 no,
change.
no. All of them are bacteria. They all contain DNA, but there are two variations. One of them is rough, and the other one is smooth. So I'm asking why the smooth one, the S3, is virulent, can cause disease, and then the R3, and the response cannot be that one of them does not contain RNA.
Okay.
I think who can help with that? I think I said it.
The one has a capsule.
Yeah, one of them is encapsulated. The S3 is encapsulated, and so it becomes virulent.
Okay.
Emmanuel Japon, you hand something.
No, hanging.
Say that with the S strain, it covers us up immune system as compared to the house strain. That does not have the protective shield around it, and else will be.
Come again. We're experiencing a terrible feed from you. So
with the S strain, it will cover itself with a polysaccharide capsule. That will protect it
from
with the house strain.
antibiotics. Yeah.
But with the house strain, that is the rough one. It is harmless due to the fact that it does not have a protective shield around it, and hence will be killed by the host immune system.
Exactly. Yeah, so it's the encapsulation that distinguishes their pathogenicity or the virulence of the strains. So let's go to Hershey and Chase experiment. And I've told you that after Griffith's experiment of transformation, they made a conclusive presumption that, well, that transforming substance must be DNA, but there is a need for further evidence to suggest that it is indeed DNA, because some scientists were still having proposition, well, genetic material could be protein, it could be RNA and all that. So they wanted to distinguish that. And in this particular case, notice that the difference between RNA and proteins are clear. There is sulfur in proteins, but there isn't sulfur in DNA. There is phosphorus in DNA, there isn't phosphorus in proteins. And so through radioactive tracing, if we label these radioactive isotopes of sulfur and phosphorus, for example, we can be able to trace which substance has been transferred to the next organism through that tracer technique. So before we even get to this experiment, Hershey and Chase, just like we did with the Grifith's experiment, one person should just give us overview of what is happening in Hershey and Chase experiments in 1915. Guys, anybody? Yeah, Dr. Emmanuel Ijapon. Dr. Ijapon.
Is there?
Emmanuel, you are speaking, but we're unable to hear you. In the absence of that, of Emmanuel, Kusi Augustine, dear doctor.
See, the experiment was done to prove that DNA was the genetic material after the Frederick Griff experiment, and then here they used viruses, and then these viruses were going in through medium, through media, like what he said, and one of them was having sulfur, and that was having phosphorus, and the sulfur indicated that it was proteins. Phosphorus is found in DNA, the phosphate group, but not in T by they are separated by species. And so the larger one go down, and then the smaller one goes up. And the bacteria were larger. So the
I
larger,
lost you, so
and
kindly
so
take it
the
again.
larger bacteria which went. Okay, so I was saying that the experiment was done to prove that DNA is the genetic material, and then here they had they, they had viruses, and then they were cultured in the two media, one with sulfur, which was found in proteins, and one is phosphorus, which is found in DNA and phosphate. And so they infected the bacteria with these viruses, and then they did centrifugation, like in centrifugation, separated by difference in densities, and so the larger one will come down, and then the smaller one in the terms of density will go up. And so when they separated the bacteria and the viruses, the larger bacteria went down, and then they were radioactive, meaning that they contained the radioactive phosphorus, and then the other one which contained sulfur went up, and so the one which contained the radioactive phosphorus was found to be that since it contained the radioactive phosphorus, then it contained the genetic material, which is DNA. And so that was how they were able to prove that DNA is the genetic material.
All right, thank you.
So I'm done.
All right, thanks. Who else? Who else? Anyone else? All right, so I think next time we will start from here, and then we'll take Avery McCroy, the McCarthy, and some other stuff. But let me have a summary of what he said.
Sir, please, I can't see the screen.
Hello. Yeah. Who is this, please? Is who? I'm having a class. Somebody is complaining she can't see the screen. Well, the screen is on.
Yeah, I can see now. I can see now.
Oh, and you've made me stop it. Let me share again. But I think we can even do away with the screen now. I'm almost done. So so let me just reflect on a basic principle. Based on radioactive tracer techniques, we can trace the movement of compounds. For example, Emmanuel Fa, we can see you sleeping. You can off your video. Based on radioactive tracer techniques, we can trace the movement of compounds that are radioactive. And examples of these radioactive compounds that have radioactive isotopes are, for example, phosphorus, sulfur. They have radioactive isotopes. This is how we do, for example, in ecological studies, where we want to determine a food chain. And so we incorporate radioactive isotopes in certain foods, and then in some grasses, for example, then all organisms that you would study and examine that have that radioactive isotope means that they fed along that food chain, and you can be able to trace that in that particular regard. So it's quite easy to trace the movement of stuff using radioactive techniques. So Hershey and Chase exploited that idea of radioactive tracing in their experiments in trying to verify what Hershey and Chase had found in their transformative experiments that suggested that the transforming substance must be DNA. And what they do is to incorporate phosphorus, that is radioisotope, that's radioactive, into the DNA of a bacteriophage, and then incorporate sulfur, which is radioactive, into the proteins of that particular bacteriophage. And if you know the molecular biology of how viruses attack, viruses are independent of, they are incapable of having independent reproduction. That's why you can't even consider them to be cells. And so all that viruses do is to hijack your cells and then inject their viral DNA into your cell. And that viral DNA that's injected into your cell hijacks your cell's nucleus and instructs the nucleus to produce couples or multiples of the viral proteins. So instead of your cell division being instructed by your own DNA in your nucleus, that instruction will now come from the viral DNA using your own cell's machinery. So that's a very smart tool of viruses hijacking system. And that's why viral infections, for example, are very difficult to treat, because it's very difficult to harm the virus without harming your cells. They attack your cell, hijack the nucleus, control everything with their viral DNA using your cell's expenditure, because they are not capable of independent reproduction. So if we know this, and a virus attacks, and in this case bacteriophage, and bacteriophages are viruses that attack bacteria, E. coli for example. If a T2 phage attacks an E. coli, and we've labeled the proteins of the T2, we've labeled the proteins, we've labeled the proteins and DNA of that T2 phage, and we know that the virus would inject its genetic material into the bacteria. At the end of the day, let's let that injection happen, and let's go and investigate from the bacteria what the virus injected. Did the virus inject its proteins, or it injected its DNA? We can tell this by doing a radioisotope analysis, whether we will find the radioisotope of the phosphorus or we will find that of the sulfur. If we find the sulfur, that means the bacteria transferred its proteins and not DNA into the bacteriophage. If we find the phosphorus, that means that the bacteria transferred its DNA and not the protein. So this is the basic principle that Hershey and Chase exploit in trying to prove what Griffiths had found in his experiments of transformation in that particular. So that's the general principle, but we'll be able to do the details next time we move and then look at Avery Macleod McCarthy, and that will be for this part of it. But let me give the opportunity for general questions, if any. If there are not, then we'll be making headways to end today's discussion. But the floor is open. And notice that when they did, they found out that it was a DNA that was transferred. Guys, any question? Or I should ask questions. Oh, because you guys want to close, that's why you're not asking any questions. So that the one who asks the question, you go and beat the person. In that case, yeah, let's just have 3 questions. If not, I'll ask questions myself. So somebody is saying they can get a recording. Fortunately, I recorded today's session, and I've always not wanted to record because some of you just miss the class and they will come for a recording. But notice that those people have been punished already. They will lose the marks for this lab because I took attendance 5 different times, and I've told you there is no way you can prove to me that you were in a class at any point in time. On 5 different occasions, you were not in a class. And all those who joined the class with some names, iPhone, this, and that, it will not be my fault that you lose marks. I'll see if I can post this recording for you guys to have access to it so that you have a second look at whatever you missed because of a disconnection or something. In the absence of... Yes, please.
Hello, sir. Sir, please. I've been hearing that an assignment has been posted into the Google Classroom that we access with a code. Please, is it true?
Okay. What post the assignment?
There's no assignment, master. What assignment?
Hey, there's no assignment. Wow.
There's no assignment.
Yo, why? Hey. Relax. Don't come and beat the girl. Let's be civil.
Oh, relax. It's not nice. It's not nice.
Hello. She was only inquiring whether there's an assignment or not. To my best of knowledge,
she doesn't have to ask anybody.
To my best of knowledge, I don't know of any assignments posted in the book.
God bless you. God bless you. Thank you, sir. God bless you.
But
okay, thank you, sir.
I would find out. Normally, Prof gives assignments to
don't find anything
any
out.
one of us. So I'll find out.
Don't find anything out. You beg.
Hey,
God bless you, sir.
Again,
Thank you very much.
I don't find out anything. Those of you doing that, be careful. Let's still bear 100 billion marks. So I'll find out if there is any assignment. I'll reiterate that to your class rep and you'll be duly informed. But it's totally in place to verify if that is the case. Because it's not just one person. Someone might have posted with an instruction from Prof, and I would not probably know about it. So don't take my response as though there is no assignment. I have to still find out. All right. So if you have any auxiliary issue, any specific issues that we did not discuss, or you want to further clarification, maybe you can contact me later. But in the absence of any direct question to what you've done here, then I think we close. I can see a few hands up. Some hands are up, but I can't really see those. Charles. Charlie.
Please, I have a question.
Okay.
Like, I just want you to send us the questions.
Okay. The question Prof promised you.
Yeah.
Hey, now I promise some question and send it. Okay. I'll remind him. And then,
thank you very much. Please make it 400, not 800.
You want 400 questions, not 800. That one,
yeah.
That one is not within your remit.
Okay.
God touches his heart. Is there any other person? That's all.
Yes.
So the class is
Thank
officially closed. Have a nice weekend. Bye bye.
Thank you. You are welcome.
Thank you,
Please, same to you. Bye bye. God bless you. God bless you.
Bye.
Thank you. Bye bye. Thank you. Thank you.
Thank you. Bye.
Someone text me. Someone text me.
Take your number.
So he gave us the assignment.
Like assignment too much.
Yeah, bro.
Oh, baby, I wanna. Hey, boy, who's your room, Anna? Ah, boy, hey, say, on my guys, boy. Hey, boy, I mean, oh, man. I did. What are you on? Are you wanting? Puma. Hey, boy, Puma on one. Oh, Papa does it. Papa does it.
Oh, I did my next 60 room, 64A.
You Billy Barazo, Mavi. Fine girl, seen
Mr. Monko
it.
Dana, move free class book no pass card.
Apa ni record the moment?